Bueno, pues buenas tardes a todos, buenos días a todos todavía. Les agradezco su participación en estos seminarios de Frontiers in Genomics online. Y if you want to share your screen, that will be fine. If you want to go ahead.
Yeah, definitely. Share
That will be
screen.
fine. So, el día de hoy tenemos al doctor Bohdan Khomtchouk, que es un bioinformático. Él trabaja en la Universidad de Chicago y es egresado de la obtuvo su doctorado en la Universidad de Miami. Después realizó un par de postdocs en California, en UCSF, como un fellow de la American Heart Association, en los laboratorios de los doctores Hosani y Asimes, que son realmente especialistas y gente que han hecho el campo de la bioinformática con la idea de construir portales que van a permitir la traslación de los datos clínicos y biológicos hacia nuevas formas de terapéutica. Entonces, con esto, con esta formación tan dirigida ya hacia la bioinformática y la terapéutica, en los últimos años el doctor ha construido un portal que está enfocado en el estudio de las enfermedades cardiovasculares y accidentes vasculares cerebrales también. Entonces, con esto en este portal es actualmente el mejor portal en donde se recoge la información genética acerca de este tipo de enfermedades y, como les digo, esto con la idea de generar, de buscar nuevos blancos terapéuticos y de generar también, pues, lo que se hace cada vez más, una medicina más personalizada en función de las variantes genéticas que cada uno de nosotros tenemos. Quisiera, pues, entonces agradecerle. I would like to thank you so much for agreeing to participate in our program and in the program online, which is a different format as we usually have. And well, thank you so much for your talk yesterday and for your talk today.
Yeah. Thank you very much for the invitation. I think I heard UCSF.
It wasn't UCSF. Okay.
Stanford University.
All right. Sorry.
40 minutes away. Yeah, like same area,
About.
San Francisco Bay Area. Yeah.
I apologize.
No, it's all good. It's all good. So, I really enjoyed giving the talk yesterday to the student seminar, and my talk today is specifically a general public seminar, and I'd like to cover sort of the most important, points and the most important take-home messages of, what I spoke about in the talk yesterday, and sort of distill down those key insights into this talk today, and show some future directions, show what it is that my lab has been working on, and also inspire hopefully sort of the next generation of students and scientists that are attending this talk today. So with that, let me just go ahead and kick off here. So when I was planning this talk, I thought long and hard about what would the title of this talk be, and for me it was actually not an easy decision because whenever I used to be when I was a kid, I used to go to Barnes and Noble and Borders Books and Music. If you remember, those stores still used to exist. People still used to go there. Barnes and Noble still very much exists, but not very many people go there. And I used to really always be drawn to the futuristic books about what's going to happen in 2050, 2100, etc. So in many ways, I remember picking up those books and reading them and being like, yeah, no, this doesn't really sound too realistic. I don't know if I believe it. But when I was thinking about the title of this talk, I realized that I can't think of anything else to name it except for Cardioinformatics in 2050, because I want to talk about future projections. I want to talk about, how I see the field, as someone who's been, actively pioneering and building this area for the last many years. where do I see things going? Where do I see the momentum, of scientists? What kind of projects are they pursuing? What research are they doing in terms of fighting heart disease with computation? Applying informatics methods to the study of cardiovascular disease. That's why I named this talk Cardioinformatics in 2050. So it's designed to show you sort of the future of how I see technology treating the #1 global killer in the world, cardiovascular disease. More people die from it every year than of cancers and any other neurodegenerative disease or any any category of mortality. Cardiovascular disease is the #1. And so the question is, how do we do something about that? What has been done about that to date, and sort of where are things going? So with that, I'm going to kick off here. Let me go ahead and put this up. There we go. All right. So the paper that we published in November 2020, actually was published in December 2019, but it finally went into citable form in November 2020, is called Cardioinformatics, the Nexus of Bioinformatics and Precision Cardiology. So this is a paper that I had started back during my time at Stanford University, which is why I made the small correction of UCSF and Stanford. I have published this paper with a couple of very interesting folks. So Tim Asimas, the senior author here, he was one of my postdoctoral advisors based in the Division of Cardiovascular Medicine in the Department of Medicine, so the Stanford University School of Medicine. Or Gozani was my other postdoctoral advisor. He was is still based in the Department of Biology at Stanford University. And then here, also co-senior Matthew Might, who was the graduate advisor, PhD advisor of DM Trang Tran, who was a co-first author with me on this work. And basically, Matt Might, if I remember correctly, he was the presidential advisor to the Obama administration and also at the beginning to the Trump administration. So very, very high up there in terms of science advisory, science policy. And so this was a really exciting piece to put out by the community, I think, to be able to illuminate, the path forward for how we see bioinformatics and computational biology methods having a direct impact on cardiovascular healthcare and the treatment of patients. And so I want to talk about what makes cardioinformatics unique and challenging, and why cardioinformatics. So research in cardiovascular disease faces unique challenges due to the many peculiarities of these diseases. One such idiosyncrasy, for example, is time scale. So for example, atherosclerotic plaques and other cardiovascular risk factors build up over an extended period of time, often over the course of many decades, which puts cardiovascular phenotypes on a complex and continuous spectrum of transition from health to disease. In other words, from disease onset to progression. And in contrast to diseases like cancer, which are characterized by rapid progression, often with a clearly delineated before and after disease state, cardiovascular pathologies such as coronary artery disease develop over an extended period of decades, beginning, for example, with atherosclerosis and manifesting variably along a spectrum from asymptomatic to stable ischemic heart disease to acute coronary artery syndrome, and then, for example, sudden cardiac death. So there is a progression. There is a trajectory here, and that trajectory can be different from patient to patient. And again, in contrast to diseases like cancer, where there's a rapid progression with a very clearly defined before and after disease state. For example, there's a stark mutational profile difference, like a stark mutational, like genomic profile difference due to somatic hypermutation. Stuff like that doesn't exist in cardiovascular disease, which makes things a lot harder to explore, and thus the need for computation to be able to pick apart those differences is critical. In general, the transition from one cardiovascular disease like hypertension to another, for example, atherosclerosis, which may gradually morph into another cardiovascular disease like coronary artery disease, which may or may not ultimately lead to a clinical episode like a heart attack or a stroke, all over the time scale of several or more decades, poses its own unique set of unique informatics challenges. And so monitoring the complex disease etiology of such a temporal progression that's influenced by a combination of genetics, so omics profiles, environment, so socioeconomics, for example, the zip code can often be as or even more important than the genetic code at predicting CVD risk. This was information that was published at Harvard many years ago. Lifestyle, so smoking, diet, for example, lipids, alcohol, et cetera. It's a very computationally challenging task and calls for new innovative data integration approaches for risk stratification and surveillance at both individual and population levels across different racial ethnic groups. And that's why cardioinformatics is unique, and that's why it's challenging. Also, to complicate matters further, cardiovascular pathologies frequently present as comorbid or multimorbid with other disease phenotypes, such as diabetes, cancer, obesity, metabolic syndrome, rheumatologic disease, et cetera. And so that complicates things even more. Innovative systems biology medicine approaches to increase the understanding of the multifactorial complex underpinnings of cardiovascular disease promise to enhance cardiovascular disease risk assessment and pave the way to tailored therapies. That's how I see it. And so you see this today in terms of, how are we treating our patients? What is the current, state of care? Where are we on the cardiovascular healthcare spectrum in terms of how far away are we from true precision cardiology? Has cardioinformatics taken a hold yet in the clinic? Things like this. And these are the questions that my lab has been exploring. And of course, at the end of the day, it's also about, a lot of people out there talk a lot about bridging the bench to bedside divide and how important that is for science, right? We have all heard of the bench to bedside divide, but how many of us have heard of the informatics to medicine divide? How do we bridge biomedical informatics to creating better medicines? And then how do we apply that to the number one global killer? In other words, cardiovascular disease. That's essentially the momentum and the spirit behind the cardioinformatics initiative that my lab has been working on. So my lab works specifically on the creation of databases. Actually, we just published a paper, New Year's Eve publication here, December 31st, 2020. It was a really nice way to say goodbye to what was a really challenging year. So happy to see this finally in print. And the title of our paper was Heart Bioportal 2.0, New Developments and Updates for Genetic Ancestry and Cardiometabolic Quantitative Traits in Diverse Human Populations. And here's a little excerpt here where I highlighted the words triple by the year 2050. So again, the title of my talk, Cardioinformatics in 2050. Here's what's coming, guys. The world's aging population, so people over the age of 65, is projected to triple by that year, which creates an impending rise in future demand for healthcare innovation within the cardiovascular disease market sector, since it is well known that age is the number one risk factor for cardiovascular disease manifestation. And so this is actually how we started the introduction of this paper. We actually led with this because it's important to recognize the impact that the healthcare system and the patients within it will experience over the next 3 decades. And so we're happy to see this paper finally, like I said, in print. This is a lot of work that we've been doing over the last many years, actually going back all the way to my time at Stanford when I published Heart Bioportal 1.0, which was published in the journal Circulation, Genomic and Precision Medicine. And I did this work in collaboration with Bob Grossman here, the senior author. He is the main PI of the Genomic Data Commons, which is the world's most advanced and the world's largest cancer data ecosystem from the NCI, from the National Cancer Institute. So Bob is a good friend and colleague of mine, and we co-wrote this paper together. It's kind of a tour de force for the field of cardiovascular disease and the role that computation plays within it. Very excited to see this out. Really encourage you to read it if you have a chance. There's another part in the introduction to the paper where I talk about the vision of Heart Bioportal. So the vision of Heart Bioportal, I said, has always been to bridge these gaps. So when I talk about gaps, I'm talking about the informatics to medicine divide gaps. By heralding in a data-driven future for cardiovascular clinical patient care, effectively building the computational supply needed to address future demand at the population levels projected in 2050 and beyond. So I'm reading you these excerpts here from this recently published paper to highlight the supply-demand curve here. And what's at stake is people's lives. And what must be done to address the supply side will be the topic of the rest of the seminar. And the paper is quite long, by the way. So here I'm talking about the Heart Bioportal 2.0 paper. That one's actually pretty manageable in size. The Cardioinformatics review article is like 20 pages. So if you don't have time to read 20 pages, I am active on Twitter and did put out a tutorial on March 4th, 2020. Here below, you see, if you click on that, you will see basically a couple of 140 character blurbs to summarize the main points of the manuscript, and talk about the importance of cardioinformatics, and sort of distill down in a couple of minutes read what is going on for the field. And also, I am pretty active on Twitter. In the bottom right is my Twitter handle, Bohdan. So if you want to follow my posts or send me a message on there, I'm fairly active. In fact, I think if you send me a message on Twitter, I'm more likely to respond than by email, because my email gets clogged up every day. So feel free to reach out. And also, as far as email is concerned, just my first name, Bohdan, at uchicago.edu. So if anyone wants to reach out after this talk to set up a time to speak privately, I'm happy to do that as well. I'm really excited about talking about some of the science that's being done, both in my lab at UChicago as well as by my colleagues in the department. Also, there was a press release that the University of Chicago put out about the cardioinformatics paper, and there is also encourage you to look at that as well, in addition to the tutorial. One thing that I will highlight in this article, which is called Research Sites on Heart Disease Discoveries Through Precision Cardiology and Bioinformatics. There is a quote that I wanted to highlight from our section chief of cardiology. So he was our department chair of cardiology. He said, "The field of cardiovascular disease has always been data and technology driven, but when it comes to taking advantage of the vast amount of information that already exists and developing a systematic approach for analyzing it, the field has fallen short. The emergence of computational biology and bioinformatics in the cardiovascular field will provide insights and potential therapeutic avenues for the treatment of cardiovascular patients." So in the talk that I gave yesterday to the undergraduate student body was specifically focused on the potential therapeutic avenues. And there we discussed how we can fight heart disease with computation to enable better cardiovascular, renal, metabolic disease drug discovery. So cardiorenal and cardiometabolic diseases present as comorbid very frequently with cardiovascular diseases. They are all part of the same system. The biology is extremely complex. So we kind of treat it from a systems biology perspective. And in fact, here at the University of Chicago, I'm based in the Institute for Genomics and Systems Biology, so the IGSB. In fact, I probably should update my my cover slide, my title slide with that information. So we are based also at the IGSB, so the Institute for Genomics and Systems Biology. We like to look at diseases not as a singular entity, but as a system within a coordinate system of other maladies. So we're looking at comorbidities, we're looking at underlying molecular mechanisms underpinning the pathophysiology of the manifestation of these diseases. We're looking at temporal trajectories, et cetera. And so we try not to leave any stones unturned and analyze biology, healthcare, medicine from a systems perspective. So again, motivation is that cardiovascular disease is the #1 global killer. As I mentioned, mortality rates in the United States, if you look at and plot them out, cardiovascular comes in way ahead of cancer, which comes in way ahead of chronic obstructive pulmonary disease, which comes ahead of diabetes. And again, diabetes, type 2, presents as comorbid very frequently with different cardiovascular disease phenotypes. And so this is an illustration this is a figure actually from our Briefings in Bioinformatics 2020 paper. And one interesting thing to note here is that by the year 2030, so less than 10 years from now, there is a projection, an estimate that almost half of the adult population will have a CVD diagnosis. which is really crazy to have so again, I'm going to say that again. By 2030, almost half of the adult population will have a cardiovascular disease diagnosis. That means that statistically speaking, between you and me, one of us is going to have, statistically speaking, a cardiovascular disease diagnosis by that year, which is, I think the World Health Organization reported on that. I have it cited in the Briefings in Bioinformatics paper. It's a really, really scary statistic, and definitely something to keep in mind as we think about the allocation of funding for research in this area. When we think about time spent in terms of a research effort, it's very important that we address this head on and address it, before 2030 actually rolls around and any of these predictions come true. Because even if it's, 1 in 3 people, it's still going to be, a tremendous amount of people. But 1 in 2 is just absolutely scary. Almost half of the adult population. So not taking kids into account here in this particular case. Congenital heart disease has its own statistics that I won't get into in this talk. So speaking of funding, right? So CVD research is, so when I say CVD, I mean cardiovascular disease. I'm just using shorthand here. It's very underfunded relative to cancer research. So if you look at NIH funding for cancer versus cardiovascular disease since the year 2008, on average, it's $500 million less per year. That's $500 million per year consistently. So this categorization was done by the NIH starting in the year 2008. Through the Research Condition and Disease Categorization System, the RCDC system. There's no data before 2008 because that's when the system started. So there's just no data that I could, mine and analyze and extract when I wrote this, Briefings in Bioinformatics paper. So we can only see what happened from 2008 onwards. Also, you'll notice that the data here stops at 2018 because 2018 is when I first submitted the Cardio-Informatics paper for review. But if you wanted to rerun this plot, actually all of the source code to generate this figure is open source on GitHub. And I will show a slide in the next couple of slides showing where exactly you can access that source code and rerun this chart and see the most up-to-date, up to 2021 version of it. Data here for this chart was downloaded from the NIH Reporter tool, so the Research Portfolio online reporting tool. Cancer, in terms of the purple line here, was cancer funding is calculated from projects, any projects being categorized as brain cancer, breast cancer, just cancer, cancer genomics, cervical cancer, colorectal cancer, HPV, basically, there's a lot of different categories: pediatric cancers, prostate cancers, uterine cancers, vaginal cancers, et cetera. So we tried to use biomedical natural language processing and text mining approaches to extract all that content and put it into a chart. And again, all that source code is publicly available on GitHub. And then cardiovascular diseases here is defined as any type of funding that was allocated towards cardiovascular research, cerebrovascular research, so strokes, for example, congenital heart disease, coronary heart disease, hypertension, pediatric cardiomyopathy, again, strokes, et cetera, et cetera. Also, we included aging, since a lot of the aging funding being doled out is actually to explore cardiovascular disease. And in fact, my postdoctoral funding as an American Heart Association postdoctoral fellow at Stanford was originally to explore the role of aging in cardiovascular disease. And I ultimately pivoted that towards creating Heart Bio Portal as a database for the broader research community to use to explore some of these things. Now, if you look at cancer versus cardiovascular disease papers that use bioinformatics methods, you see that there's a much smaller computational footprint in cardiovascular disease relative to cancer. So here we are looking at primary research articles. We are excluding review articles. So we are only looking at primary research, and we are text mining PubMed. Specifically, we are looking at something called medical subject heading terms, so MESH tags in PubMed, and we're looking at all the MESH terms for bioinformatics and for genomics. And since all of the queries are based on a manual MESH catalog, these two you see here, it stops at 2018. Again, it's because that's when the paper was originally submitted. The 2018 tallies will lag behind the true volume of publication. So for the most up-to-date results, for the most up-to-date charts, just rerun the code in the GitHub, and that's actually what I'm gonna, I think, show you on the next slide here. So here's a Zenodo for keeping a DOI and version control over our GitHub account for Informatics. So it's all the data and all the source code release for the paper. It's here to facilitate reproducibility in order to power the visualizations like the ones that I presented on the previous few slides. And basically, again, the code is open source. I don't even remember if I have a license on this, so you can do whatever you want with the code. You could play around with it. You could modify it. You could explore different parameters that I didn't explore in the paper itself to ask new questions about, the state of cardiovascular disease research funding or cancer research funding, et cetera. I mean, heck, you could even modify it for, whatever disease area that you're personally interested in. As long as you cite, the paper, like I'm happy. So check it out. Like it's on GitHub, and yeah, it was a lot of work to put this together. And by the way, all the code is written in the R programming language. So we originally were entertaining Python, but ultimately chose to use R because of some really nice packages in the ecosystem that we leverage to make these analyses happen. Another visualization in the paper, we looked at the bioinformatics footprint in cardiovascular disease and found that it's actually small in general, not just relative to cancer. So there's a very small percentage of total cardiovascular disease papers being published currently that include bioinformatics techniques or methodologies as part of the study design. So likewise, there are far more CVD papers released annually than bioinformatics papers in any domain area, not just in CVD, highlighting sort of the tremendous momentum and the volume of new research results being published in the field, in the cardiovascular disease field. And again, here we are once again looking at mesh terms for bioinformatics and genomics, and then we are categorizing, and in some cases, we are also manually curating content. So if there's one thing that you got from all those slides, sort of the main take-home message is that we need more bioinformatics in cardiovascular disease. And obviously that means funding for it. So how do we get there? And in my opinion, it's a question of changing the culture. So we know that cardiovascular disease is the #1 global killer. It takes the lives of more than 17 million people per year. We discussed that by 2030, almost half of the adult population will have a cardiovascular disease diagnosis of some sort. That doesn't mean you'll have a heart attack or a stroke. No, it means that you might have hypertension, you might have atherosclerosis because your coronary artery calcification score is high, your CAC score. You might have something else, but the fact is, the projection is that almost half of the adult population will have something. And so we know that right now there's a funding disparity at the NIH in terms of cardiovascular disease versus cancer research. And specifically, there's been a $500 million difference that's been consistent year in and year out. And it's not getting any better. And in fact, if you know anything about the NIH, right, we have the NHLBI, which is the main center, National Heart, Lung, and Blood Institute, that, the main institutional center that funds cardiovascular disease research, but it also uses the same pot of funding to fund, research in sleep disorders, research in lung, research in blood disorders, et cetera. So there's a much smaller piece of the pie. So I've always asked the question to myself, if there's an NCI, if there's a National Cancer Institute, why isn't there an NHI, a National Heart Institute, right? Why doesn't that exist? Because if you look at the American Heart Association, for example, the amount of money that those guys have for research allocation, for research funding is much smaller than NIH money. And in general, most of their grants are much smaller in scope and in total funding. So that creates huge disparities within the US. Like, I can't speak very much about the European system. They have their own organizations. They have their own organizations for this. But in the US, there's definitely a huge disparity in funding, which creates, of course, inevitably, inexorably creates research disparities because there's no money, relatively speaking, to oncology research. So as I mentioned, the bioinformatics footprint in cardiovascular disease is modest at best relative to cancer. And the hypothesis that I have is that by expanding this footprint with improved data science, so bioinformatics techniques, I think that will ultimately improve healthcare, because we've seen this happen for precision oncology. We've seen this be the case for precision oncology, that expanding the data science footprint for cancer research has ultimately improved healthcare. There's no doubts about that. Read the statistics if you don't believe it. Look at all the large academic medical centers that are rolling out these treatments. Look at the morbidity, mortality of these patients across different healthcare systems, and you will see year in, year out that we have been beating cancer back more and more and more and more. And the question is, when are we going to start doing this for cardiovascular disease and the management and treatment of patients with these conditions? So assuming that cardioinformatics can impact healthcare, the question is, where do we start? So I remember when I first started my postdoc at Stanford, prior to taking a faculty position here at UChicago, I asked myself this question a lot. And, it was a really hard question to answer because, there's so many things that you can do, right? When you have carte blanche to work on, almost anything you want to work on, you're sort of, you just completed your PhD, your area of interest is cardiovascular healthcare. You want to apply computation towards fighting it. How do you decide what specific, project to apply yourself to? And because I was at Stanford, and Stanford has always been very, entrepreneurial. Like everyone there is interested in technology transfer, taking academic research and spinning it off into the markets, checking the market opportunities, the competitive landscape that exists for commercializing technologies. When you work on software, like it or not, you're going to have to want to talk about users. So I said, let's start with users, because, more users, in Bay Area, Silicon Valley land, right? More users equals more value. So in other words, build something people want. Actually, that's a quote from Y Combinator, which is one of the big startup incubators in Silicon Valley. Their motto is build something that people want. So something people want to use. And so I wanted to, my objective was to capture the entire broader cardiovascular disease genetics research community, which is a relatively small, but also rapidly growing user base. So obviously, we're not talking about Facebook's 2 billion users here or some other such scale, right? We're talking about a niche community in a specific medical subspecialty, cardiology, and specifically people who are interested in the genetics of cardiovascular disease. And right, that's like a funnel within the funnel within the funnel. So obviously you're going to have a much smaller number of users, but it's not really about the users as much as it's about the market share of those users. So are you capturing like 100% of, the number of users who are in your field, who all know about and use the resource that you've set out to develop? And so when I was thinking about developing such a resource, I asked myself, well, which cancer resources have many users? So of course I knew about the Genomic Data Commons, actually, so you saw the Heart Bioportal 2.0 paper that I published with Robert Grossman, as senior author. So he's the main PI of the Genomic Data Commons. And you see he wrote that paper back in 2016. It was published in the New England Journal of Medicine here in the bottom right, toward a shared vision for cancer genomic data. That's where they discussed the functionality and the utility of the National Cancer Institute's Genomic Data Commons platform. Of course, I knew about, the existence of cBioportal. So, sort of the biggest, resource for looking at complex genomic data sets relevant to oncology and creating intuitive visualizations out of them. I knew about all of these. And you see, they're all highly cited, which, they provide intuitive visualization and analysis of large-scale cancer genomics data sets for the broader research community of cancer genomics users. And so they are accessible to biologists with no programming expertise, which means that a really large amount of people in the oncology community can leverage and take advantage of this resource and be able to use it to plan their next set of experiments, to understand how exactly to take the next steps for creating a hypothesis that they would like to test, writing grants, checking hypotheses that they believe may be true, and then looking at those questions from a data-driven point of view by performing various queries in cBioportal, et cetera. So we followed this model for Heart Bioportal, specifically not for cancer genomics users, but for cardiovascular disease genomics users. And that's why we named it HeartBio Portal, so that people who heard the name, if they have heard of CBio Portal, it would immediately create those, cognitive associations with an existing resource, so that they know, oh, there's an equivalent of that for cardiovascular disease data. That's pretty cool. And these two resources, right, the Genomic Data Commons and CBio Portal, they also capture effectively 100% of the market share of cancer genomic researchers out there. And so we wanted to do the same thing with HeartBio Portal. And so when we published this paper in April 2019 in the journal Circulation Genomic and Precision Medicine, as you can see here, I published it with my two postdoc advisors, Tim Asimis and Ora Gozani. I remember when we submitted this and then we got our reviews back, I remember Tim, who is a fellow of the American Heart Association, he told me that he has never seen a paper be accepted this quickly. In fact, like he said that in his entire career, he's never seen reviews that good. So the first set of reviews that we got were basically like, it's almost like we wrote them ourselves or something. It was crazy. The reception was phenomenal for this, and we were very happy because that, as a postdoc, I was, in the faculty job market at the time, and getting a paper out there quickly was always a relief for me, so that I don't have to put in my CV, under review or in revisions or something like that. Right? It's published. It's done. So this was a very nice thing. By the way, the HeartBio Portal 2.0 paper that we submitted also was accepted very quickly. We actually we submitted it on October 25th, and it was accepted on December 15th. So like less than 2 months, which is very, very fast for most journals. And the students sitting in on this talk will know that things don't happen that quickly in terms of reviewing and receiving commentary, feedback, reviews from your peer reviewers. Like sometimes they take 2 months just to get back to you. And this was really, really a surprise for us as well, seeing such quick turnaround with this resource. I guess that the people that were reviewing HeartBio Portal either already knew about HeartBio Portal and were active users, or we just got lucky. I'm not sure. It's, sometimes it's better to be lucky than good, but sometimes it's good to be good and to be lucky. And, when we were creating HeartBio Portal, we wanted to create something good, something that the whole community can take advantage of and find utility in, right? Is this useful to me is sort of the main question that I remember my postdoc advisor, one of them, Ora Gozani, used to tell me, you have to ask yourself that question. Would your user base consider this so useful that they keep using it and they tell their friends? That's sort of like how I approached the philosophy behind the software development of this. And part of that philosophy was user interfaces and design, right? I wanted to make it very appealing for a user to come to the site and make searches. So for example, if you go to the homepage of HeartBio Portal, it's really minimalistic, and less is more, right? There's one search bar, you type stuff in and you hit search. Unlike CBio Portal, which is actually, you're getting better now, but back when I was creating HeartBio Portal, it was really finicky, it was slow. The user interface was very confusing. They were adding stuff willy-nilly here and there, like in places that, biologists like my advisor just couldn't keep up with all the latest features. And so out of the gate, I said to myself, I want HeartBio Portal to be easy to use and a pleasure to use. Like, even if you want to see more data in it, you're going to still come back to HeartBio Portal because you just love coming back to it. It gives you, it has that ergonomic kind of, user experience that you expect when you use good software. And so software engineering and, the principles behind how we, set out to build the product roadmap and all of its respective features was really, really, really important to us. And thankfully, we nailed that. In fact, when we send out Survey Monkeys for HeartBio Portal, all of our users say that the site is very easy to use. It's very easy to navigate. And I don't think anyone's mentioned any improvements yet that they would like to see to the user interface design itself. Also, we have a Twitter account for HeartBio Portal. So we try to keep it fairly active. And we discuss various new features, new data coming into HeartBio Portal. Sometimes we issue Survey Monkeys. Sometimes we highlight other people's work. and how Heart Bioportal can help with exploring that work further. So as we bring new data sets in, as we bring new studies into the portal and make them searchable and accessible to others, we like to talk about that research. And, you know, now that Heart Bioportal is so large in scope, every time I add a new data set in, every time I add a new paper in, it's no longer just an N of 1 that I'm adding. It's no longer just a single paper. It's sort of like with this much data already in the system, every new paper that we add expands the richness of all the other papers and all the other data sets that we've already added, because together the total is greater than the sum of its parts. And that's the beauty of creating data commons databases like Heart Bioportal, is that together data is more than data separately, right? Breaking down those data silos is the number one most important thing that you can do in order to drive biological insight. And on our end, we take care of all the manual curation of that data. We take care of the harmonization of that data. And if you visit our frequently asked questions section in Heart Bioportal, so there's in the menu of our homepage, there's you don't see it here on the slide, but if you go to our FAQ section, you'll see how we do that harmonization, how we do that curation. And we even have a section for users who want a different type of curation or a different type of harmonization to get in touch with us. And we can work with them to curate and harmonize and analyze the data using, for instance, specific bioinformatics pipelines that they want to see. So taken together, how am I personally contributing to cardioinformatics? So my lab is writing scalable and secure engineering infrastructure to power the world's cardiovascular disease genetics genomics data. We're talking about publicly available data at the moment, so we are not talking about controlled access data sets, things that you have to apply for data access requests to, that you have to, send them to data access committees. We're not talking about that. We're talking about publicly available data sets that are de-identified and that they're okay to be public. Although we do have plans, and I'll show you this in the supplementary slides, about how we would like to interoperate Heart Bioportal with resources that provide access to controlled access data sets. So that's how I'm contributing to cardioinformatics. Now the question that you have to ask yourself from this talk is, if you want, how can you contribute to cardioinformatics? Clearly, you don't have to work on databases like I do. There's many different ongoing areas of research, all of which we covered within our cardioinformatics paper. For example, you could work on cardiovascular disease risk stratification, so things like polygenic risk scores. You could work on AI and machine learning for cardiovascular disease prediction modeling. You could work on parsing and clinical phenomapping of cardiovascular disease phenotypes. So for example, heart failure. Heart failure is it has huge heterogeneity. It's a very complex disorder. And so can you parse it out into finer, more granular subtypes of heart failure that ultimately impact patient care because you're ultimately capable of categorizing patients in a more precise, precision medicine way. You could work on discovering rare and causal variants across different demographics. In fact, that's something that I'm writing grants for right now here at UChicago. Specifically, I'm looking at HIV-associated cardiovascular disease, which is a very exciting area of research where, if you know anything about HIV, it's that it's manageable now. It's chronic, but it's manageable. People are not dying from AIDS anymore because, HIV is manageable. And so the question is, what are they dying from? So these days, people who are undergoing antiretroviral therapies are living long, productive lives, but they are ultimately dying from cardiovascular disease because all these antiretroviral therapies are causing all kinds of different systemic inflammation. And inflammation is a killer for cardiovascular disease pathophysiology. And so you have things like that. You have fields like cardio-oncology, which explore how do we manage, now that we've started to successfully manage patients' cancers, how do we keep them from developing coronary artery disease or subclinical atherosclerosis? How do we manage those conditions? Because all these chemotherapeutics, they are again causing widespread whole body systemic inflammation in people's arteries. And so it's leading to cardiovascular disease. There's so much fascinating systems biology there that there is room for everybody in cardioinformatics. You could even do things like explore structural variation in cardiovascular disease. I saw on Twitter how just recently someone discovered a gigantic structural variant that's like tens of thousands of base pairs long, not like a SNP, like a single base pair mutation, but like a huge chunk. And somebody found in a very well-known gene, PCSK9, that's like one of the most well-known, most well-studied genes that's been clinically validated in so many different clinical indications. Like we thought we knew everything there is to know about PCSK9, and we were wrong, because there was a huge structural variant that was recently discovered in the Icelandic population. And it was like, well, in my Twitter network, like it was sort of a sensation of, again, another reason if you're interested in cardioinformatics and you want to follow me and look at, stuff that I'm reading and following and interested in, please go ahead and do that. Again, my Twitter handle on the bottom right is Bohdan. Also, another area that I'm very interested in is natural language processing. So biomedical, text processing, text mining. As you saw in some of those visualizations, those figures, those charts that I showed you from the Briefings in Bioinformatics paper where we looked at funding, how did we analyze all that data? We mined that data from PubMed using a variety of different, accession identifiers like the medical subject heading tags, the mesh tags. And in general, bioNLP is a fascinating area that's compared to an area like imaging, for example, that has a lot of people working, that has a lot of progress. BioNLP, and especially its applications to cardiovascular disease, is actually very, very, very much smaller footprint. And in fact, the only big place that I know that really does a lot of great research in this area is University of Colorado. I believe the Denver campus specifically. So again, please read our cardioinformatics paper for a full description of each of these topics. These are all different ongoing areas of research. In my case, I work on databases and I work on developing resources for everybody. But there are 101 different things that you can work on within this field, and there's room for everyone to make an impact. Now I have a set of supplementary slides and screenshots of several different HeartBio portal features. I'm going to pause here and not go in that direction because I don't know how many questions I'm going to receive. It's been 41 minutes since I've started giving this talk. Maybe some of these supplementary slides will be able to answer some of the questions. But if you would like, I can keep going. But let's see how many questions in this general public seminar I have, and then we will pick it up if we need to fill in the hour. Thank you, everyone.
Hold on, there might be.
And if there's not, then I will just pick up because I have at least 15 to 20 minutes more on the supplementary slides that people might be interested in.
Right.
I just want to leave room.
There's a long question on the chat.
Oh yeah, chat works. Let's see.
Right.
Oh, okay. Yeah, great. Let me see.
My question is, so go to the third paragraph. So my question is, given the information we have, is there any bioinformatic methodology that could help to identify long non-coding RNA's function could be more relevant in cardiovascular diseases?
That's actually a really good question. I've been thinking about long non-coding RNAs ever since 2017. So I think I have something to say about this question. So I don't think there's any novel methodology that needs to be created to study this. So a little bit of background about me. When I did my PhD, I was working in an epigenetics lab with a huge long non-coding RNA component. I was working with Klaus Wallstedt at the University of Miami School of Medicine, and his entire career was put into epigenetics and long non-coding RNA. And I had published, articles in the Journal of the American Heart Association and other such journals exploring, like some of these epigenetics questions. And one thing that kept coming up over and over again is how do we, like this question says, how do we identify what specific long non-coding RNAs, like function could be more relevant to cardiovascular diseases? There's been a lot of great research published over the last couple of years since 2016-17 when I first started thinking about this. I would first defer to go and look there to see the current state of the art. I know that it is a field that doesn't get as much attention as it deserves. I know that there are labs out there that specifically focus on long non-coding RNAs. I know that in HeartBio Portal, you can actually, so there are, when you look at these visualizations, you can there's a data download button. So you press that button and you get these tidy data downloads, these CSV files where you can actually subset the results, and long non-coding RNAs are definitely something that is present in there. You may have to, in the gene names, you may have to put those gene names through one of those online web tools that tell you like which genes are relevant to long non-coding RNA. So you may have to do an extra sort of additional step, but that's the point of these CSV files. We want you to use them to perform additional secondary analysis on these publicly available, curated, harmonized data sets. And so I really, really highly encourage you to explore that and see what you can find, because I think that it is a very ripe area of research that's currently doesn't get as much attention as it deserves. Most people looking at long non-coding RNAs are looking at it for autism, looking at it for different neurodegenerative diseases. Some are looking at it for cancer, but almost nobody's doing it for cardiovascular. So that's a really good research area. Let's see.
And then
next question.
There's another one.
In the
so maybe you want to read it?
Yeah, I'll read it. Yeah. So in the clinical setting, there's still this stigma surrounding cardiovascular diseases related to weight, stigma, bias. Do you think that by harnessing the existing multi-omic data plus presenting it in a physician-friendly way, you'll be able to add in the process of breaking down CVD and weight stigma? That's a great question. Every time, a patient goes to the doctor, the first thing that the doctor tells them is, I got advice for you, lose some weight. And, body mass index, right, BMI, it's one of those things that, is highly linked with cardiovascular disease manifestation, right, obesity, et cetera. I don't think any kind of multi-omics data will have a tremendous impact on that. I think, so for example, like obesity is a very, very, very vibrant area of research. And there's many companies, like Novo Nordisk, for example, and Big Pharma that are looking to tackle, the therapeutic avenues in this. And everyone's, metabolism is different person to person. And so someone who is obese, they're not necessarily obese because they're undisciplined and won't stop eating. It's possibly because they have a metabolic, disorder of some kind. And so in those kinds of cases, yes, the doctor, if multi-omics goes into the clinic, at a large scale, yes, the doctor is going to take that into account when they're telling you to lose some weight, because maybe you're not able to lose some weight because, you need, some therapeutic, intervention to help you in doing so. And it's not like those therapeutic interventions, yet exist at a large scale. Many of them are, like still in clinical trials, et cetera. And so I think that the answer to that question is sort of yes and sort of no, because also it depends where you're getting treated, right? So in the United States, for example, right, are you being treated at a large academic medical healthcare system like UChicago that attracts the world's top doctors, or are you getting treated in some place in rural, don't want to name a state to not offend anyone, right? Question is, are you in an urban, very popular metropolis that attracts a lot of talent and everyone's trying to place there as a resident, be a doctor there? Are you somewhere very rural where no one wants to work there, right? So clinical care like really differs, when you go from different geographies to different geographies. Yeah, I think that harnessing existing multi-omic data and presenting it, also presenting it to a physician in a friendly way. Physicians, they're not necessarily trained to be able to interpret genomic medicine data. Now that is changing, but again, that is only changing in certain really top institutions. Like how do we train tomorrow's medical doctors to be able to provide care for their patients and take their genetic information into account? So yeah, that's a really tough question to answer, and it has a lot of angles and a lot of sides to it. Okay, next question. What are your perspectives in this area? Are health institutions, whether private or public, ready for this challenge? And what are the specific areas that need improvement in your opinion? That's a very open-ended question. Yeah, I mean, I think it's not about being ready. I think it's a necessity, right? I mean, if you want to get something done, like unless you're really getting forced into it, you're probably going to keep procrastinating. It's like with deadlines, right? If you have a deadline, you're gonna work towards getting it in by that deadline. There's like research that says that like most people submit their applications like within a week of the deadline. If you look at grant mechanisms that don't have a deadline, for example, like the NSF has a data infrastructure capacity grant for data commons that doesn't have a deadline. Most people like actually don't submit as many applications there because it doesn't have a deadline, so they're not motivated. Now, what is that deadline in cardiovascular healthcare? So that deadline is that 2050 projection, where by the year 2050, the aging population over the age of 65 will triple, right? Creating an impending rise in the demand for cardiovascular healthcare innovation, an expansion also of the system to be able to handle that many people, and also being able to provide them the treatment that they need in order to live longer, happier lives with, greater quality of life. Like those are all very important things to consider, and that's kind of the deadline. The deadline is time. The more we wait and the more we just sit around and, not allocate the proper research funding to investigate, areas within cardioinformatics, the more we're just, sitting ducks and just waiting for cardiovascular disease to come and take us all, right? That's ultimately what it is. I mean, statistically speaking, anyone on this call is most likely to die from some kind of cardiovascular disease thing, heart attacks or strokes, and the trajectory leading up to that. So we have to change, right? It's almost like with global warming, like with climate change. Like we have to take steps to fix what we've been doing and change our ways in order to make the world that we ourselves live in a better place for ourselves and for our health and well-being. That's kind of how I see it. Hopefully that answers what is a pretty open-ended question, but hopefully I was able to shine some of my insight on that specific point. Okay, next question. Cardiovascular diseases are complicated, especially since many environmental variables must be taken into account. But how do you face the problem of measuring lifestyle conditions such as diet, smoking, or sedentarism? Oh yeah, so those things are typically taken into account in the electronic medical health records. So, people, physicians, doctor, they take, clinical notes. Some of them are actually handwritten. So they're now digitizing this, especially in large academic medical centers like UChicago. And by digitizing it, we're able to take that into account and put that into our AI machine learning predictive models to look at data at a larger scale. So that's how we take those environmental variables into account. So cardiovascular disease is a mix of nature and nurture, right? Nature in terms of your genetics, nurture in terms of how well you take care of yourself. Do you smoke? Do you eat like crap? Do you not exercise at all, right? Sedentarism, like all of these things, come together. Also socioeconomic status, also pollution, right? Like, are you in an environment where there's a lot of pollution in the air? There have been many very interesting studies published by American Heart Association journals like Circulation that have explored the role of pollution at a large scale. These are all questions that, you have that nature-nurture balance. And from a genetic side, from the nature balance, there are people that have genetic variants that confer cardioprotective effects on them. So for example, we've all heard of the people who can smoke 2 packs or 3 packs of cigarettes a day and they live to be 99 and they're good. Like they never die of a heart attack. Heck, they don't even get lung cancer. How does that happen, right? This is genetics. There are, understanding the genetic landscape of what types of variants in what types of genes under what conditions confer cardioprotective effects on people is actually very critical to understand, and the reason why resources like Heart Bioportal exist to investigate cardiovascular disease genetics. Because if we can understand that, then we can understand how to treat our patients better. And so actually my first paper published in graduate school in the Journal of the American Heart Association was specifically on the role of cardioprotective effects. In that case for ischemic preconditioning, which is a slightly different thing, but cardioprotective effects is still a very, very hot topic. I mean, you see biotech companies in Boston like Verve Therapeutics that are trying to use CRISPR to engineer in variants that confer cardioprotective effects on patients so that they're not dying of, coronary artery disease and heart attack and stroke. So essentially we're looking at how do we bring about those changes through genetic engineering now. So still very hot topic with a CRISPR flavor now, but definitely something to keep in mind as we move forward. Hopefully that answers that question. Next question. Hello, I have a question. Yesterday you mentioned that 40% of clinical trial failure are because no clear links are made between the target and the disease. So my question is, how did people used to link a target with a disease, and how does this approach cause the failure of a drug? In most cases, they either got lucky, this is most cases, in other cases they pivoted and repurposed the drug towards another clinical indication. So it didn't work for the disease that they thought it would work for, but it worked for something else. I mean, for example, Viagra, right, was supposed to be for cardiovascular, things, and it was turned out to be for something else. There's a lot of different, like drug discovery is not a linear path. It's a very windy road, and there's a lot of stuff that goes on behind the scenes that you don't see, in the R&D phase of, performing systematic, target identification and prioritization of which targets do we go for, and then can we, engineer, a small molecule or an antibody or some other modality to target that specific, target that we've established is the one to go for. In terms of how do people used to link it, again, there's multiple types of approaches, computational ones. I don't have the time to go into all the details, but this is something that, is covered in detail in any papers that look at, computational drug discovery. Also, there's another question here. Yeah, I'll get to that in a minute. How does this approach cause the failure of a drug? Well, yeah, because the drug, for instance, like even if a drug does target, the targeting is spot on, there could be also toxicity efficacy effects. So one problematic thing with cardiovascular disease drug discovery is that things get to phase 3 clinical trials and then they fail because the drug is, there's some kind of toxicity efficacy issue at a larger population scale, and things go sideways and the drug never gets FDA approval, and you've tanked a decade, sometimes more, billions of dollars into its development, and it just doesn't happen. This is actually the reason why many venture capitalists, investors in the biotech space, are very cautious and wary of, getting into investing in this space, because compared to like something like cancer, it's much more risky as an area of biotech investing. But yeah, it's complex biology, right? But the opportunities are huge. Like, for example, you saw last year how Bristol Myers Squibb acquired MyoKardia for $13 billion because they had a drug that was FDA approved for treating symptomatic obstructive hypertrophic cardiomyopathy, which afflicts like 5 million people, I believe, in the US per year, which is not that big of a market share, but it highlights this, like how valuable, how scarce good cardiovascular disease drugs, assets, are in the market, and how much, big pharma is willing to pay to acquire them. So huge opportunities, high risk, high reward is what I would call it, right? It's risky to get into the space from a drug discovery point of view, but if you nail it, like, you're rich. And you've just touched the lives of so many patients that were used to be on beta blockers for hypertrophic cardiomyopathy. Like beta blockers, they're just not that effective, and I can get into that all day for patient care. I'm actually really interested in how that works, and I have a lot of clinician friends who tell me a lot of really scary stories about this. But anyway, okay, next question. In your paper, you mentioned that it's very hard to fully define the effect of a single gene within cardiovascular diseases, because there are too many at a time for any to have a big impact. My question is, what are the contemporary methodologies that have the sensitivity to detect these candidate genes and assess their impact without risking too big of a false discovery rate? Oh, this is a good question. So you'll notice in the paper that most of the methods covered are actually genetic association methods, so they're GWAS methods. GWAS is great, but GWAS, a lot of geneticists think that, just because we find an association, and how do we like take that to the next level in terms of like drug discovery and therapeutics, right? And the more patients we recruit to our GWAS cohorts, the more statistical power we have to see signal that we didn't see 5 years ago because that many patients didn't exist. So let's imagine cardioinformatics in 2050. What's going to happen? There's going to be sample sizes, patient cohorts that are much bigger. And so we're going to discover a lot more signal. And now what do we do with all that signal? How do we take that towards the next step of, drug discovery and actually bridging that informatics to medicine divide that I've been talking about over and over again during this talk? These are all open questions. I think personally that Mendelian randomization is something that's very promising right now for looking at those kinds of, GWAS data sets. So looking at causal inference, not just association, right? Like, how do we know that this SNP in this gene is actually causal for that disease phenotype, as opposed to just being associated with that disease phenotype? So innovation in the statistical methodology, in the computational bioinformatics, computational biology methods that we use to analyze these data sets. That's how I think you would approach the, what you said in terms of without risking too big of a false discovery rate. Another thing is polygenic risk scores. They have been shown now to be very effective in the clinic, way better than traditional risk score models. And this is still a subject of debate between many people, like in my Twitter network, who's right, who's wrong, and everyone's throwing tomatoes at each other. Like, no, my risk model is better than your risk model. No. You need to look at, if you're interested in the algorithm space for this, look at two things. Look at Mendelian randomization, look at polygenic risk scores, and look at what they've been doing with existing GWAS data. GWAS is useful. I'm not kicking GWAS. GWAS is critical, but GWAS isn't enough to make those causal links. And so that's where, instrumental variable analysis techniques like Mendelian randomization methods have come into the fore to be able to answer some of these, questions of how do we actually causally link a specific mutation to a specific disease phenotype. Next question. Do you think that knowing who is more predisposed on having a cardiovascular disease could have an economic implication who will be given medical insurance? Oh, that's a great question. So this goes back to like the Genetic Non-Discrimination Act of 2008, I think, right? GINA, right? Like of course, like you can't discriminate, against a patient if you know that they have, a specific genetic condition. Now, there's really not that many genetic conditions in the cardiovascular disease space that would be as debilitating as, for example, Huntington's disease, right? Where if there's a mutation in a specific gene for Huntington's, a person's going to be a paraplegic by the time they turn 40 or older, right? There's really, there are certain things that, confer a great risk on a patient, but those are typically, things associated with those cardioprotective or actually lack thereof effects that we discussed earlier. I don't think there's going to be, if done right. Again, there are entire people who work on the ethical concerns of genetic data, that work on the privacy aspects of not identifying these data sets, right? This is the reason why it's so important, is that we don't want that information to ever be used in the wrong way. And so I think that it's a double-edged sword as we get more advanced in terms of having genetics come into the clinic, into the cardiology clinic. The more we have to start asking those questions and thinking about those things. And actually, in my Heart Bioportal 2.0 paper, the one published in Database on December 31st of last year, I talk a little bit about that as well, about how, you know, genetic recommendations have even made their way into clinical practice guidelines. So the actual written, you know, tenets that you know, cardiologists use when treating their patients. So those clinical practice guidelines now very often incorporate genetic recommendations. So it's entering the scene more and more and more. It's definitely here to stay. It's only going to grow in the clinic, and like you said, that's something that's very important, you know, to think through. How do we protect our patients, their privacy, et cetera, and the ethical implications of, you know, healthcare insurance and those kinds of questions. But I am certainly not the best person to talk about that. That is for genetic counselors. That is for clinicians that are specialized in seeing patients. All I see is a keyboard and a computer screen and a ton of data that I have access to, and I analyze that data. So if you're interested in those kinds of things, you need to be talking to the people working at the front lines of, you know, talking to the patients, right? I'm the guy who's trying to bridge the, you know, informatics to medicine divide. They're the ones who are bridging the bench to bedside divide. We have, you know, division of labor in the cardioinformatics space. So next question. Some heart disease happens very fast. So instead drug development to cure, how much are the efforts to prevent the disease? Is there a biomarker done with bioinformatics? I'm not sure I understand that question. Let me think. I mean, there's definitely biomarkers, right? So that's actually the whole point of doing, you know, all this cardioinformatics stuff is you want to pinpoint, you know, what are, for example, the causal genetic variants that are leading to the manifestation of a specific, you know, cardiovascular disease phenotype, and looking at that clinical indication, what are, you know, what is the mutational landscape in general? You know, what are the driver mutations? What are the passenger mutations, like using oncology terminology here? You know, what is sort of the main driver, you know, causal variant that is leading to the progression, deterioration of the patient? Cardiomyopathy is actually really fascinating as an area to study some of these questions. And that's why I was mentioning, like, beta blockers, they're not that like they do not actually like you know, like help in terms of like slowing down the deterioration of the patients in hypertrophic obstructive cardiomyopathy. When you say some heart disease happens very fast, I think maybe you're referencing sudden cardiac death. That is a very, very active area of research from a genetics angle. There are entire publications focused on the genetics of sudden cardiac death. And most of the people doing that are actually, I think, based in Australia. And actually one of them, I believe, is the editor of the Circulation, Genomic, and Precision Medicine journal that I publish HeartBio Portal version 1.0 in. Chris Simsarian, also very active on Twitter, also highly encourage you to follow his account. I follow his account, learn a lot from some of his tweets. So yeah, hopefully that answers that question. Sorry if I didn't understand all the finer points of the question, but hopefully I give some insights. And there are no more questions. So with that being said, I think we are at the 1 hour, well, actually 1 hour and 7 minutes.
We're a little bit over.
Okay, great. So I'm glad I stopped.
So we really thank you very much for this talk. And well, I think you really are, you know, in a field that is emerging. And as you say, you are bridging, right, between the informatics and the clinics field, so that new avenues are known now.
Yeah, well, I try.
No, let's try well.
Thank you very much for the invitation. This was a pleasure.
Okay, thank you so much for being with us. And I guess you have already one of our students that was working with you this summer, right?
Yep, that's right.
And so that's how we got to connect you, actually. So
yep,
thank you so much for being with us.
Thank you. Okay. I really enjoyed this. Thank you very
much.
Have
a good time. Thank you.